A very good afternoon to all. So, technical poll has been launched. Quickly answer me through poll. My screen is visible and my audio is clear. Okay. So, today we are going to discuss molecular basis of inheritance part 1. Okay. So, today we are going to discuss the structure of polynucleotide, structure of DNA, discoveries regarding the DNA, and the criteria for the properties of the genetic material and replication. And the second part, we are going to discuss the transcription, translation, and the regulation of gene. So let's start with part 1, that the molecular basis of inheritance, part 1. First, we are going to discuss the structure of the polynucleotide chain. As you know that nucleic acid, the cells, they have nucleic acid, which is a genetic material, and the nucleic acid could either be RNA or the DNA. RNA as a genetic material is mostly found in case of viruses. Okay, and the cells, they have a DNA as a genetic material. So we know that the nucleic acid, they have a monomeric unit which is known as nucleotide. So the monomeric unit of nucleic acid is nucleotide. And DNA is a polymer of deoxyribonucleotide. You know that the nucleotide is consist of three things. One is phosphate group, sugar, and nitrogenous bases. These three, that phosphate, sugar, and nitrogenous bases, they combine and they form the nucleotide. Sugar and the nitrogenous base, they form nucleoside. And there are two types of nitrogenous bases are present. There's purine and pyrimidine. Purine are double ring-like structure. They have double ring. Adenine and guanine are purine. Pyrimidine, they are single ring. Example is cytosine, uracil, and thymine. DNA, they have thymine, whereas RNA, they have uracil instead of thymine. And in DNA, I told you they have a deoxyribose sugar. And RNA, they have ribose sugar. Please remember this. And these are the polynucleotide chain, where the sugar and phosphate, they form the backbone of the polynucleotide chain. And on the inward, these nitrogenous bases are present. And these nitrogenous bases, they are connected to each other by the hydrogen bonding. So adenine will pair with thymine with the 2 hydrogen bond, and cytosine will pair with the guanine with the 3 hydrogen bond. Always remember the proportion of the purine and pyrimidine are equal or constant in the DNA structure. That is why we get a uniform. You can see that there's a uniformity in the distance between the two strands of the DNA because the proportion of the purine and pyrimidine are constant. Moving next, I told you that the nitrogenous bases and the pentose sugar, they form the nucleoside. And when the nucleoside are attached with phosphate, they form the nucleotide. This is a sugar. These are the nitrogenous bases. The linkage between the sugar and the nitrogenous bases is known as a glycosidic linkage. Remember this linkage. And the two nitrogenous bases, they are connected by the hydrogen bonding. And the sugar are attached to the phosphate by the phosphoester bond. So this phosphoester bond and this phosphoester bond, they together form the phosphodiester bond. And also remember that the strands of the DNA has 2 strands. It is double-stranded. Okay, it's a double helical structure. So the 2 strands, they are anti-polarity. They show anti-parallel polarity. For example, this strand is having a 3' end and the other is having a 5' end. Similarly, here The one end is the 5' end and the other end is the 3' end. Here you can see the phosphate is attached to the 5 carbon of the sugar, and this phosphate is free. That is why we call this end as the 5' end. Here, the sugar at the 3rd carbon of the sugar, the free hydroxyl group is present. So this end is known as a 3' end. Moving next is the structure of DNA. DNA was first discovered by the Frederick Miescher, and he called the DNA as nuclein. Later, the X-ray diffraction data was proposed by Wilkins and Franklin, and based on these data, James Watson and Francis Crick, they proposed the double helical structure or model of the DNA. According to this model, the DNA has 2 polynucleotide chains. These are the polynucleotide chains. So DNA has 2 polynucleotide chains, and these polynucleotide chain, they have sugar and phosphate as a backbone, and the nitrogen bases, these nitrogenous bases, they are projected inward. You can see these nitrogenous bases, they are projected inward. They also said that these strands of DNA, they show anti-polarity or opposite polarity, like 5' to 3'. This strand is showing the 3' end and 5' end. So they are showing the anti-parallel polarity. Second, they said that these nitrogenous bases, they are joined or they are connected by the hydrogen bonding. The adenine, they pair with thymine by the 2 hydrogen bonding, and cytosine, they pair with guanine with the 3 hydrogen bonding. And I told you that the proportion of the purine and pyrimidine, or the proportion of adenine, will be equal to the proportion of thymine, and proportion of cytosine will be equal to the proportion of guanine, because of which we get a uniform distance between the 2 strands of the DNA. Next one is that the 2 strands of DNA, they are coiled in a right-handed fashion, and the pitch of the DNA, or the pitch of the helix, is 3.4 nanometer, and the distance between the 2 nitrogenous bases is 0.34 nanometer. We know that the DNA is a double helical structure. So these bases, they are stacked over one another. This base pair, they stacked over one another, because of which it provides a stability. And we know that these nitrogenous bases, they are connected by the hydrogen bonding. The bond between the 2 nitrogenous bases is hydrogen bonding. So this hydrogen bonding also imparts the stability to the helical structure of the DNA. Next one is the Ergin-Chargaff rule. Ergin-Chargaff said that the proportion of adenine will be equal to the proportion of thymine, and the proportion of guanine will be equal to the proportion of cytosine. Okay, so adenine, so the ratio between the adenine, thymine, guanine, and cytosine are constant and equal to 1. For example, here you can see this adenine and guanine, they are purine, thymine and cytosine, they are pyrimidine. So the proportion of purine is equal to proportion of pyrimidine. That is why we are getting the uniform distance or uniformity in the distance between the 2 strands of the DNA. The length of the DNA is 2.2 meter in case of human, and the dimension is of the typical nucleus is 10 to power minus 6 meter. You can see that the typical nucleus size is very small, and the length of the DNA is very large, which is 2.2 meter. Now we have to package this DNA in such a way that it can fit into this nucleus. So in case of eukaryote, we know that DNA is negatively charged. And why DNA is negatively charged? Because DNA has phosphate. Because of the presence of the phosphate group, the DNA is negatively charged. And this negatively charged DNA, they wrap around the histone proteins. These are the histone proteins. So DNA will wrap around this histone protein. These histone proteins, they are positively charged. They are basic proteins. And these proteins, they are rich in lysine and arginine amino acid. Remember this, that histone protein are rich in lysine and arginine. There are 4 types of histone proteins are present. There's H3, H4, H2B, and H2A. They all are present in, they all together form the histone octamer. Octamer means 8. So there are 2 copies of each histones are present. The H2A will be 2, H2B will be 2, H3 will be 2, H4 will be 2. So all the histones, they are present they're present in 2 copies and they form the histone octamer. This is the histone octamer. Okay, and the DNA will be wrapped around this histone octamer and form a structure. That structure is known as a nucleosome. Remember that the typical nucleosome contains 200 base pair of DNA helix. Remember this. It's important that a typical nucleosome contains 200 base pair of DNA helix. One more important point. 2 nucleosomes, they are connected to each other by the linker DNA. This is a linker DNA, this is nucleosome number 1, this is nucleosome number 2, and 2 nucleosomes, they are connected by the linker DNA. And this linker DNA is associated with the linker histone. Linker histone is present at the point. This is the linker histone, which is the H1 histone. So H1 histone, also known as the linker histone, and it is present at the position where the DNA entered the nucleosome and DNA exit from the nucleosome. So please remember this. It's very important that the linker histone is not the part of nucleosome. Remember this. This H1 histone is not the part of nucleosome. Okay. This H1 histone or the linker histone is associated with the linker DNA. Nucleosome only contains the DNA which is wrapped around it, plus the histone octamer. And histone octamer consists of H2A, H2B, H3, and H4. And these nucleosomes, they constitute the repeating unit of structure in a nucleus called chromatin. These are the nucleosome. There's nucleosome 1, 2, and 3. These are the nucleosome. And the structure, these nucleosome, the structure is known as chromatin. The entire structure is known as chromatin. And in chromatin, the nucleosome, they appears like a bead on strings. And further packaging of these chromatin, or the higher level of packaging of chromatin, requires another set of proteins which are known as a non-histone chromosomal protein. In this image, you can see this is a DNA. Now this DNA is packaged into a nucleosome. And now they form a structure like a bead on the strings in the chromatin. This chromatin, they're further coiled, then they form a structure like chromatin fibers. Then the chromatin fiber, they further coil, they form a loop-like structure. And at the cell division, we see chromosome. And when the chromatin, they condense and get compacted, they form a structure which is the chromosome. And these chromosomes will be seen clearly during the metaphase stage of the cell division. Remember this. Chromatin, they are generally of 2 types. The euchromatin or heterochromatin. Euchromatin, which is the chromatin which is loosely packed, lightly stained, and transcriptionally active. Whereas heterochromatin are densely packed chromatin. Because they're densely packed, so they appear dark in color. So they are darkly stained. And they're transcriptionally inactive. This is the heterochromatin. You can see the DNA or the chromatin here, they are tightly packed. Because they are tightly packed, so the transcriptional machinery, they cannot assess the DNA. And when the transcription machinery, they cannot assess the DNA, so the transcription will not take place. So this region or the heterochromatin is transcriptionally inactive. Whereas euchromatin, you can see in euchromatin, the chromatin are loosely packed and the DNA are exposed. Whereas the DNA, because the chromatin is loosely packed, so the transcription machinery can easily assess the DNA and the transcription can take place. So euchromatin are transcriptionally active. There one more important point, this acetyl group. These acetyl group, they get attached to the histone. And when this acetyl group, they get attached to the histones, some modification occurs in histone, because of which the histone loses the affinity for the DNA. When the histone loses the affinity for the DNA, so that means the DNA will get loosed up, or the chromatin will get loosely packed. It will result in the loose packaging of the chromatin, because of which the transcription machinery can easily assess the DNA and it lead to the transcription. So euchromatin, always remember that euchromatin, they are transcriptionally active and heterochromatin are transcriptionally inactive. Moving next is the transforming principle or the transformation. So transformation experiment was performed by Frederick Griffith in 1928. And he worked on Streptococcus pneumonia, which is a bacteria, and this causes a disease which is known as pneumonia. So he took two strains, this S strain and R strain. This S strain is virulent in nature, and this R strain is non-virulent in nature. This S strain, why it is virulent in nature? Because it can synthesize this mucus polysaccharide coat. Whereas the R strain, they cannot synthesize the mucus polysaccharide coat, because of which they appear rough. They have this rough appearance. Okay, so what Griffith did, he injected the mice with the R strain, which is non-virulent. He observed that the mice is alive. The mice survived. Next, he injected the mice with the smooth strain, which is the S strain. And we know that S strain is virulent in nature because it has the mucus polysaccharide coat. So this S strain leads to the death of the mice. Third, he heat killed this S strain and injected into the mice, and he observed that the mice survived. In the fourth, what he did, he injected the R strain, live R strain, and the heat kill H strain together into the mice, and he observed that the mice die. So from this experiment, he concluded that there's something which is transferring from the S strain into the R strain that has converted this R strain into the S strain. That means that has allowed this R strain or the R bacteria to synthesize the mucus polysaccharide coat, because of which it becomes virulent in nature and causes the death of the mice. But Griffith, he could not explain the biochemical nature of the transforming principle. So next experiment was done by Avery, MacLeod, and McCarthy, and they explained the biochemical nature of the transforming principle, which Griffith could not explain. So what Avery, MacLeod, and McCarthy did, they purified the biochemicals, which are protein, DNA, and RNA, from the heat-killed S strain. So this test tube contains all the biochemicals which are purified from this heat-killed S strain, that's DNA, protein, and RNA. First, what they did, they subjected the solution in the test tube with the proteinase enzyme. So proteinase will act on the protein and they will degrade all the protein. So when they pour the solution into the culture containing the R strain, they observe that the S cell appear. Okay, that means the R strain, they're converting or they're transforming into the S strain. Next, what they did, now they treated the solution with the RNase enzyme. Now the RNAs will act on RNA and they will degrade the RNA. When they pour the solution into the culture containing the R cell, they observed that again the S cell appear. In the third tube, what they did, now they use DNAs. Now DNAs will act on DNA and they will degrade the DNA. So when he poured this culture into the R cell, he observed that no S cell appear because the DNA is already degraded by the DNase enzyme. And we know that the DNA is the biomolecule which is transforming from the S strain into the R strain that has converted this R strain into the S strain and causes the death of the mice. So they observed that the no S cell appear in this case. So from this experiment, it was concluded that the biochemical nature of the transforming principle is the DNA. But many scientists were not convinced, or many biologists were not convinced that the DNA is the genetic material. So the ultimate experiment was done by Hershey and Chase. And through this experiment, they proved that the DNA is the genetic material, or the DNA is the genetic material which is transferred from one generation to next. So they worked on bacteriophages. Bacteriophages are the viruses that infect the bacteria. So they grew these bacteriophages under two media. One medium contained the S35 radioactively labeled sulfur. Another media contained the P32, which is radioactively labeled phosphorus. We know that the DNA, it consists of sugar, phosphate, and nitrogenous bases, right? Let's say nucleotide. We know that the nucleotide, it consists of sugar, phosphate, and nitrogenous bases. So phosphate is a part of nucleotide, and nucleotide is a part of nucleic acid. So phosphate, which is radioactively labeled, will get incorporated into the viral DNA. Why? Because the phosphate is present in nucleotide, and if we label this for radioactively label the phosphate, this phosphate will get incorporated into the viral DNA. Similarly, there's sulfur, and we know that there's one amino acid we call it cysteine. This amino acid contains sulfur, and because this is an amino acid, so it's a part of protein, right? So this radioactively labeled sulfur will get incorporated in the protein coat of the virus. So first step of this experiment is the infection. In the infection, these bacteriophages, they attached on the surface of the bacteria cell. This is the bacterial cell. So all the bacteriophages, they get attached on the surface of the bacteria cell, and they will incorporate the viral DNA into the host DNA, that is the bacteria. So you can see in this image, all the viral DNA has entered into the bacterial cell. Second step is the blending. In the blending, when the blending takes place, the bacteriophages, or the protein coat of the bacteriophages, they get detached from the bacterial cell or the host cell. Third step is the transfugation. When the transfugation is done, it was observed that the S35, or the radioactive sulfur, is observed in the supernatant. And the palette, which consists of the bacterial cell, they show radioactive P32. That is, the radioactive phosphorus is observed in case of the bacterial cell. So from this experiment, it was concluded that it is the DNA which is transferring from the bacteriophages into the bacteria. So DNA is the genetic material, not the protein. Okay, because we are getting the radioactive sulfur in the supernatant, and we are getting the radioactive phosphorus in the bacterial cell. So that means the viral DNA is transferred from the virus into the bacteria. So it was concluded that the DNA is the genetic material. Next one is the properties of the genetic material. There are some criteria for the genetic material. Number one is the replication. So we know that DNA and RNA, they can replicate, but protein cannot replicate. So that means the protein is not the genetic material. Second, it should be stable chemically and structurally. We know that RNA is less stable as compared to DNA. Why? Because DNA is double-stranded. Second, on the DNA has deoxyribose sugar. You can see in this image at the second carbon of the sugar, there's a hydrogen is present in case of DNA. That is why we call it a deoxyribose sugar. Whereas in RNA, we have ribose sugar. At the second position of the sugar, there's hydroxyl group is present. Because of the presence of the hydroxyl group at the second carbon of the sugar, it imparts a highly reactive nature to the RNA. So RNA is more reactive as compared to DNA because of the presence of the hydroxyl group at the second position of the sugar. Second, the DNA, they have thymine, and RNA, they have uracil. So thymine is more stable as compared to uracil. And from the experiment, it was also concluded that the DNA is stable, is structurally stable, because when this S bacteria was heat killed, so the bacteria die, but the genetic material remain intact, or it does not get destroyed. Why? Because when the heat is given to the bacteria, the denaturation of bacteria, when the heat is given to the DNA, the DNA get denatured. Denaturation means the strands of the DNA, they get separated. But when the appropriate condition arises, because we know that the DNA strands, they are complementary in nature. So when the appropriate condition arises, these strands of the DNA, they come together again, or the realigning with takes place. So the DNA, they are structurally stable. They are less reactive as compared to RNA, whereas RNA, they are structurally unstable and they are highly reactive. Another important point is that DNA, they show slow changes. Okay, whereas RNA, they show very fast changes because RNA is highly reactive, and when because the RNA is highly reactive, so it shows a high rate of the mutation. So the organism which are having the RNA as a genetic material, they evolve very faster. For example, viruses. The viruses, they generally have RNA as the genetic material. So the viruses, they show a high rate of mutation and they show the high rate of evolution also. So DNA is chemically less reactive and structurally more stable as compared to RNA. First, that it should express itself in the form of Mendelian characters. Next one is the RNA wall. We know that RNA was the first genetic material. Please remember this, that first genetic material was RNA. And RNA, it can use as a genetic material as well as a catalyst. Why it is using as a catalyst? Because it is highly reactive. And because it is highly reactive, so RNA is unstable. So DNA has evolved from RNA with some chemical modification that makes it more stable. And DNA is more stable because it is a double-stranded structure. It has complementary base pairing because the two strands, they are complement to each other, because of which they can resist the changes by evolving a repair mechanism. Moving next is the replication. Replication is semi-conservative in nature. Semi means half. So if this is the parental strand, So this is the parent. One strand will remain conserved and another strand will be newly synthesized, or the daughter strand. So this strand is conserved. It's a parental strand. And this one is the new strand. So it is semi-conservative in nature, whereas the one strand will remain conserved and the other strand will be synthesized. And they can be easily synthesized because we know that both the strands, they are complementary to each other. Michelson and Stahl, they performed the experiment in 1958 on E. coli. And they also concluded that the DNA is semi-conservative in nature. What they did, they grow the E. coli in a medium containing ammonium chloride. But here the nitrogen is N15, which is a heavy nitrogen isotope. So they grow this E. coli in a medium containing N15. After that, they transfer this E. coli into a medium containing N14. And N14 is a light nitrogen isotope. Now they extracted the DNA from the culture periodically at different interval of time. When they extracted the DNA from the culture, after 20 minutes, they observed the hybrid or intermediate density. Because the one strand, this red strand, they are the parental strand which remain conserved, and these green strand, they are the newly synthesized strands. So the conservation of DNA is semi-conservative. So the replication of DNA is semi-conservative in nature. Next, they extracted the DNA after 40 minutes from the culture. When they extracted the DNA from the culture after 40 minutes, they observed the light band and the hybrid. Because now these green will also act as a parental strand, and the newly strand will be synthesized because both the strands, they are complementary to each other. So we'll get light band as well as the hybrid. And very similar experiment was also performed by Taylor on Vicia faba. But they, in E. coli, Michelson and Stahl, they use N14 and N15 isotope. Remember this. But Taylor, on Vicia faba, they use radioactive thymine. And Vicia faba is a eukaryote, E. coli is a prokaryote. Remember that Michelson and Stahl, they performed the experiment on E. coli, and E. coli is a prokaryote. Whereas Taylor, they performed the experiment on Vicia faba, and Vicia faba is eukaryote. So they use radioactive thymine. And this experiment also proved that the DNA in chromosome, they replicate semi-conservatively. So the replication is semi-conservative in nature. We know that the replication cannot occur at any random place. There are some origins are present. There are specific sites are present. And these specific sites are known as the origin of replication. And the unit of DNA replication is known as replicon. So replicon is consist of the origin, the site where the DNA replication start, and then replication fork is formed, and the replication proceed bidirectionally and reaches to a terminate point. So this entire unit is known as a replicon. So prokaryotes, they have single replicon. And we also call it a circular replicon. That prokaryotes, they have a single replicon and circular replicon, because the DNA in case of prokaryote is circular closed DNA. Whereas eukaryotes, they have multiple replicons. They are multi-repliconic, and they have linear replicons because eukaryote, they have linear DNA. We know that a replication requires certain enzymes. The first enzyme is the helicase enzyme. Role of the helicase is in the unwinding of the DNA, or to separate the two strands of the DNA. And this enzyme requires ATP. Remember this. This is a helicase enzyme. You can see this triangular shape. This is a helicase enzyme, and this helicase enzyme, it's helping in the unwinding of the DNA, or separating the two strands of the DNA so that the replication can take place. Second protein is the SSBP protein. We know that once the strands they get separated, they have the tendency to come back to their original position or to come back together because they're complementary in nature. So to prevent that, there's a SSB protein are present, which is a single stranded binding protein, which binds these the strands which are separated by the helicase enzyme. This SSBP protein bind to these strands and they prevent the reannealing of the strand. Third one is the primase enzyme. We know that the DNA polymerase, they cannot initiate the replication on its own. They cannot initiate the synthesis of the new strands of the DNA on its own. They require a primer. And for the synthesis of primer, there's an enzyme is required, which is known as primase. So this primase enzyme will synthesize a primer, which is having a nucleotide less than 15. And when this primer is synthesized, now the DNA polymerase will come. The DNA polymerase is also known as a DNA dependent DNA polymerase. Why it is DNA dependent? Because it is using the DNA as a template and starts synthesizing the new strand. So these DNA polymerase, they will start adding the nucleotide to the newly synthesized strand, and they form a polynucleotide chain. This DNA polymerase, they have high polymerization efficiency. For example, in case of E. coli, in E. coli, the DNA polymerase has a polymerization efficiency of 2000 base pair per second. Like in 1 second, it can add 2000 base pair. So they have a high polymerization efficiency. And along with that, they also have a high accuracy. High accuracy means they there's a less chances of error. Okay. And also remember that we know that these nucleotides, they're triphosphate in nature. They're deoxyribonucleoside triphosphate. So they're triphosphate in nature. So when the incoming nucleotide are added, the terminal phosphate is removed. So when the phosphate group is removed, that means the bond are breaking, the energy is released. So this energy is utilized by the DNA polymerase for polymerization. So the nucleotide, it is performing two functions. It is providing the energy for the polymerization, and along with that, it also acting as a substrate. Always remember, very, very important, that the direction of replication always occur in 5' to 3' direction. Remember this, the direction of the replication is always 5' to 3'. I told you that at the 3' end, there's a free hydroxyl group are present so that the incoming nucleotide can come and attach. So it is the 3' end where the incoming nucleotide are coming and keep on adding. So the direction of the synthesis is always 5' to 3'. Now, we know that we cannot separate the entire DNA in just one go because it requires a lot of energy. So we always open the small portion of the DNA, and it leads to the formation of the replication fork. This is the replication fork. We know that the DNA, they have opposite polarity. This strand is 3' to 5', and this is 5' to 3'. They're having an opposite polarity. Now this strand, the one strand synthesis is the leading and another strand synthesis is the lagging. Because we know that the direction of the DNA replication is 5' to 3'. So here the replication is taking place in 5' to 3' direction, and it leads to a continuous synthesis or the formation of a continuous strand, which is also known as a leading strand. But in this direction, you can see that because the synthesis takes place in 5' to 3' direction, there's a lagging fragments are formed, and these lagging fragments are known as the Okazaki fragments because it was first observed by the scientist Okazaki, so they are named as the Okazaki fragments. And these fragments, they are later joined by the enzyme which is known as a DNA ligase. Now let's discuss the question. Question number 1 says, with respect to nucleosome, which of the following statement is incorrect? They're asking about the incorrect statement. Number 1 is nucleosome are seen as bead on string under the electron microscope. Second, DNA is wrapped around the positively charged histone octamer to form nucleosome. Third is nucleosome is the repeating unit of chromatin. Fourth, nucleosome contain 120 base pair of DNA helix. Quickly answer what you think, what will be the correct option. Very good. This is the histone octamer. So DNA will wrap around this histone octamer and they form a structure which is known as nucleosome. We know that DNA is negatively charged, right? And histone are positively charged. And these repeating unit of nucleosome, they lead to a formation of a structure which is known as chromatin. So statement option 1 says the nucleosome are seen as bead on string under the light microscope. Yes, the nucleosome, they appeared as a bead on string under the electron microscope. Second one, the DNA is wrapped around the positively charged histone octamer to form nucleosome. Yes, DNA is wrapped around these positively charged histone octamer to form a nucleosome. Third, say nucleosome is a repeating unit in chromatin. Yes, the nucleosome, they are repeating unit. This is the chromatin. This is the chromatin. These are the nucleosome. So nucleosome is the repeating unit of chromatin. Yes, true. Nucleosome contain 120 base pair of DNA helix. No, they contain 200 base pair of DNA helix. I told you this. So they're asking about, with respect to nucleosome, which of the following statement is incorrect? Is number 4th. Moving next, one of the strand of double-stranded DNA has base composition as follows: 15% adenine, 15% thymine, 40% guanine, 30% cytosine. What will be the percentage of these bases in the complementary strands? Quickly answer what you think, what will be the correct option. Please apply Arvind Chargaff rule here. Then you can easily answer this. Very good, students. And the question is given like 15% adenine. 15%, that means percent that out of 100, 15 are adenine. So I can write it at adenine are 15, 15 are thymine, 40 are guanine, and 30 are cytosine, right? If you add this, this will equal to 30 and this will equal to 70. So total they will give 100, right? That is why we are writing that out of 100, 15 are adenine, 15 are thymine, 40 are guanine, and 30 are cytosine. We know that adenine will pair with thymine, right? So whatever the amount of adenine, that would be the same amount of thymine, because the proportion of adenine will be equal to the proportion of thymine. We know that the thymine will pair with adenine, right? So if there's 15 thymine, there would be 15 adenine. Guanine will pair with cytosine. If there are 40 guanine, there will be 40 cytosine. cytosine will pair with guanine. If here is 30 cytosine, so here will be a 30 guanine. So 15 thymine, adenine 15, cytosine is 40, and guanine are 30. 15 adenine, 15 thymine, 30 guanine, 40% is cytosine. So the correct option is 4. I hope it is clear to everyone. Now moving next. Read the following statement, choose the set of correct statement. A, euchromatin is loosely packed chromatin. B, heterochromatin is transcriptionally active. C, histone octamer is wrapped by negatively charged DNA in nucleosome. D, histones are rich in lysine and arginine. E is a typically nucleosome contains 400 base pair of DNA helix. So they're asking about the choose the correct statement. Quickly answer what you think, what will be the correct option for this question. Very good, student. First says that euchromatin is loosely packed chromatin. Yes, loosely packed chromatin is euchromatin, and dark and densely packed chromatin is heterochromatin. And euchromatin, they're transcriptionally active and they're lightly stained. Heterochromatin are transcriptionally inactive and they are darkly stained. So euchromatin, they're lightly stained, heterochromatin are darkly stained. Euchromatin transcriptionally active, heterochromatin transcriptionally inactive. Remember this. These are very important points. So statement 1 says that euchromatin is loosely packed chromatin. It is correct. So A is correct. B say heterochromatin is transcriptionally active. No, it is transcriptionally inactive. C says the histone octamer is wrapped by negatively charged DNA nucleosome. Yes. Because DNA is negatively charged, so they wrapped around the positively charged histone octamer to form nucleosome. D says the histones are rich in lysine and arginine. Yes, histones are rich in basic amino acids like lysine and arginine. So D is correct. A typical nucleosome contains 400 base pair of DNA helix. No, they contain 200 base pair of DNA helix. So E is wrong and B is wrong. So A, C, and D are correct. So option 1 is correct. A, C, D only. I hope it is clear to everyone. Now moving next, given below are two statements. Statement number 1 says the DNA polymerase catalyzes the polymerization only in one direction, that is 5' to 3'. Statement 2 says during replication of DNA, on one strand the replication is continuous, while on other strand it is discontinuous. In the light of the above statement, choose the correct answer from the option given below. What do you think, what will be the correct option? Statement 1 is saying that the DNA polymerase catalyze the polymerization only in one direction, that is 5' to 3'. That is absolutely correct. I told you that the replication only takes place in 5' to 3' direction. That means the synthesis of the new strand will always take in the direction that is 5' to 3'. Very good, Shishni. And the statement 2 says that the replication, that during replication of DNA, on one strand, the replication is continuous. Yes, we are getting the leading strand because the replication is continuous. On the other strand, the replication is discontinuous. That is why we are getting the lagging strand. We also call it as Okazaki fragments. So both statement 1 and 2 are correct. So correct option is 1. DNA replication is semi-conservative in nature, was experimentally proved in eukaryote by. They're asking about the eukaryote. Option 1 says MacLeod and McCarthy. Option 2 says Michelson and Stahl. Option 3 says Taylor and his colleagues. Option 4 says Hershey and Chase. What do you think? What will be the correct option? They're asking that DNA replication is semi-conservative in nature, was experimentally proved in eukaryotes. We know that the DNA replication is semi-conservative. If this is the parental strand, so this is the parental and this is the newly synthesized strand. So one parental strand will remain conserved, act as a template for the synthesis of the new strand, which is the daughter strand. Very good, Shishni. So the DNA replication is semi-conservative in nature, was experimentally proved in eukaryotes by Taylor and his colleagues. They worked on Vicia faba. Remember, and they used radioactive thymine. Michelson and Stahl, they work on E. coli. And they use N14 and N15 isotope, nitrogen isotope. Remember this. Question is asking about the eukaryote. So Taylor and his colleagues, they work on Vicia faba. And Michelson and Stahl, they work on E. coli. And E. coli is a prokaryote. Question number 6 says, which of the following experiments of Frederick Griffith resulted in the discovery of bacterial transformation? Option 1 says S strain heat killed plus R strain live injected into the mice and the mice die. Second is S strain injected into the mice and mice died. Third is R strain injected into mice and mice lived. Fourth is S strain heat kill injected into mice and mice alive. Which one of the following experiment of the Frederick Griffith resulted in the discovery of bacterial transformation? Which step? They're asking about that which step the Griffith very good, Logesh. That is number 1. Because when he injected the S strain and the heat kill S strain and the live R strain into the mice, he observed that the mice die because of the transformation, because of the transforming principle, that something is transferred from this heat killed S strain that has converted this R strain, which is non-virulent, into virulent, and it causes the death of the mice. Very good, Shishni. Moving next. The final proof for DNA as a genetic material came from the experiment of dash. One is Hershey Chase, second is Avery MacLeod McCarthy, third is Hargovind Khurana, fourth is Griffith. What do you think? What will be the correct option for this question? Very good, Shishni. Very good. So the final proof for the DNA as a genetic material came from the experiment of Hershey and Chase. They worked on bacteriophages and bacteria. They labeled the bacteria with P32 and S35. They observed the radioactive phosphorus in the bacterial cell. because the DNA is transferred from the virus into the bacterial cell. And this DNA has radioactively labeled P32. So Hershey and Chase, they concluded that the DNA is the genetic material which is transferred from the virus into the bacterial cell. Avery, MacLeod, and McCarthy, they explain the biochemical nature of the transforming principle, that the biochemical nature of the transforming principle was DNA, but many scientists were not convinced. Griffith, he performed the transformation experiment. Question 8 says that if the DNA contains sulfur instead of phosphorus and the protein contained phosphorus instead of sulfur, what would have been the outcome of the Hershey and Chase experiment? 1, non-radioactive sulfur in bacterial cell. 2, both radioactive sulfur and phosphorus in bacterial cell. 3 is radioactive sulfur in bacterial cell. And 4th is radioactive phosphorus in bacterial cell. Please read the question carefully. They're saying if the DNA contains sulfur instead of phosphorus, so that means the DNA would be radioactively labeled with sulfur. Because the DNA contains sulfur, if DNA contains sulfur instead of phosphorus. So that means the DNA, they have S, radioactive sulfur, and protein contain phosphorus instead of sulfur. So the protein, they're having the phosphorus. So radioactive phosphorus now will present in protein. This was the question. Now they have reversed the situation. Okay. Then what would be the outcome of the Hershey and Chase experiment? If the DNA is containing the sulfur, so that means the radioactive. We will observe the radioactive sulfur in the bacterial cell. Yes, very good, Shishni. I hope it is clear now. Please read the question carefully. Now they have reversed the situation. They said that the DNA contains sulfur instead of phosphorus and the protein contains phosphorus instead of sulfur. In the original experiment, we are getting the radioactive phosphorus in the bacterial cell because we know that the phosphorus is a part of nucleotide, and nucleotide, they are the monomeric unit of nucleic acid, and DNA is the nucleic acid. That is why we are getting the radioactive phosphorus in the bacterial cell. But now this question, in the question, they have said that now you have reversed the situation. Now the DNA contains sulfur instead of phosphorus. So the radioactive sulfur would be present in the DNA. So that means the radioactive sulfur would be observed in the bacterial cell. So the correct option is 3. I hope it is clear. Thank you all for your participation. It's already 1. We are winding up the class. Thank you all. See you in the next class and have a nice day.